Hey everyone, welcome back to Developmental Biology. In this lecture, we're moving on past fertilization and we are on to cleavage. I'm having a very difficult time not making a dad joke right now. I will spare you, just trying to jump right out of me. So cleavage is the next phase after fertilization, where the fertilized egg that was fertilized by the sperm, obviously, starts to undergo duplication of the cell, right? And so we start getting these patterns of how the cell's duplicating and making this kind of early ball of cells. And so, like a mortula, we'll kind of get into some of that stuff too. So this is, again, my chat GPT work here. Always got to remember to put on my laser pointer. So this is actually a decent representation. I don't like how it looks like kind of cell, like it's like cleavageception, where there's like this looks like a ball of cells that's making up a larger ball of cells. But imagine that each one of these is a cell. And so it kind of forms this little ball that will start to differentiate, and we'll make a blastula and a blastocle, and then etc. We'll go through these phases, but decent representation. But let's get into it a little more here. So this I'm going to kind of continually refer to as throughout our course to kind of orient us. This is, so if we look at evo-devo, so evolutionary development, most of these phases up till right about birth here, a lot of these phases of development are dang near identical between a frog and a human and all these different animals. And we'll see some comparative pictures coming up. But there is a lot of similarities. And so we'll use this to orient it because it's very similar to what humans are up to birth. Like, obviously there's no larval stages for humans or metamorphosis or anything like that. But so far we've done gametogenesis, right? So we've talked about the creation of egg and sperm. We've talked about the fertilization process, and now once you have fertilization, we get to this cleavage process, right? And so we can kind of see as we're progressing around here, we start with a simple division of the cell in two, then we get a morula stage, which is usually like 1632 cell stage. We'll develop into a blastula, then we'll move into gastrulation in future lectures. Right, let's move on. So this is kind of a early stage of cleavage, right? Oh, sorry, I put my pointer over here. So one thing to note that you'll see throughout these pictures is that during these events of cleavage, the number of cells grows exponentially, right? So it's constantly doubling. However, the volume of the embryo itself does not. So as it's doubling, it's not doubling in size exponentially, but the number of cells will continue to double. So as we see here, this is the 8-cell stage roughly, right? Because we see 4, but it's also 3-dimensional, so there's like 4 behind it. And if we go to the next picture here, this is the same size cell or embryo. So like the zoom is the same, right? But what you can see is that we have a whole bunch more cells, right? So we're much further along towards like a blastocle type stage here, or blastula. And so while this replication's happening, these cells are getting smaller, or at least making up a smaller percentage of the overall embryo. So during this development here, we can still see that. This is much later. This is towards kind of gastrulation. But we get this development of this dorsal blastopore lip. So 180 degrees from the point of sperm entry of the female egg is where this blastopore and the blastopore lip forms. So this is a small hole in the embryo that eventually becomes the dorsal side of the organisms. Remember, dorsal, like dolphin, dorsal fin, that's your backside, or your literal back of the organism. So there's also, we'll get into kind of these migrations of cells. So in organisms where cells migrate to form different tissue, which we'll see, they migrate through this blastopore lip to gain entry to the inside of the embryo, and that's where your organs develop, obviously, on the inside. And the stuff that remains on the outside turns into the ectoderm. We call that layer the ectoderm. So we have the ectoderm, which will eventually develop into the epidermis, so your skin, hair, stuff like that. Your brains and your nerves. That's all part that originates from the ectoderm tissue. Then we have the endoderm, which will be the lining of the gut or the GI tract and your respiratory system. So kind of everything in your lungs and gut. And then you have the mesoderm, which is kind of your precursor to your connective tissue, your muscles, your heart, your skeleton, gonads, and your kidneys, and etc. So kind of everything that's not your lungs and your gut in this general area, right inside. Well, your muscles, I guess, are everywhere too. But okay, so those are your three: ectoderm. Remember, that's your outside skin, brains, nerves. Endoderm, your inside, your gut and your respiratory tract. And then your mesoderm, which is your connective tissue, so your bones, your muscle, blood, heart, gonads, kidneys, etc. So then towards the end of this, we get organogenesis. So this is after gastrulation. So organogenesis begins when the cells of the more dorsal region, so this area, right, your back, when the cells of the most dorsal region of the mesoderm form a rod of cells called the notochord, and then it stretches across. And so this is the beginning of the neural tube, which begins your spinal cord, your nervous system, etc. So we're going to get more into this stuff later, but I just want to kind of show the progression of cleavage to actual gastrulation and organogenesis. And so then we get to this kind of somewhat final stage here. So we see the neural tube. We start to get these the somites in the brain, the forebrain. You can kind of see this is starting to look kind of like a tadpole, where we have the mouth opening here. Whoops, I'm not pointing the right finger. So we get the forebrain, the eyes, the mouth, kind of the brain up here, the somites, which will start to develop into some of your vertebrate and prey and things along that nature. And then lastly, we get our organism. So this was I don't know if this is a zebrafish, I believe, but a fully formed organism. So cleavage has some different patterns. And so cells in the cleavage stage are called blastomeres. So if they're in this stage of cleavage, we call them blastomeres. And we have these different patterns. We have isolecithal or less isolecithal. These are like difficult for me to pronounce out loud. So excuse me when I mispronounce most of them. So this is like a radial cleavage pattern. And then we have spiral cleavage. And a lot of these all fall under holoblastic. So this is a complete cleavage pattern. So you can see that each one is like an individual cell, right? And then we have that's compared to down here, which is like incomplete cleavage. You can see that these cell walls are starting to form, but they're not like complete cell divisions. So then the pattern that they do. So isolecithal, this pattern, as you can see, it goes kind of these oblong, but doubling in like layers almost. We have spiral cleavage, where they kind of duplicate along an axis. We have bilateral cleavage, where everything's just in these two halves here, like a left and a right half, bilateral, two halves. And then this rotational cleavage, this is more of what mammals and nematodes do. And so we end up with this internal cavity here. As you can see, like flatworms and mollusks and things like that, they don't need to have an internal cavity, right? Because they don't have like internal organs. Well, at least not to the extent that like mammals do. And so this pattern is a little bit different. Mesolecithal. This is for amphibians. And then we have the meroblastic. So these are the incomplete, like I said before. So these are some more mollusks. We have two different types of mollusks here. So the cephalopod mollusks, right? So this is like octopi and squid and stuff, and cuttlefish and things of that nature. Discordial cleavage, so it's where we have like budding, almost, I'd call it, where you have like the big original one, and then we have all these little tiny cells. So instead of splitting in half like we have up at the top here, we have just kind of like little buds coming off. And so this is for fish, reptiles, and birds. And then centrolithical. So this is, we'll get into syncytial and specification and things like that. So this is where, like on the outside of the egg, we get a formation of all these little tiny cells, and where there's a bunch of different nuclei that get kind of distributed. We'll get into that a little bit more. So those are the different types of cleavage. In this slide, the handout here, I've also broken them down bigger. So if you do need to make notes or whatever, you have these handouts. I have some listed here for some of these terms. So the echinoderms, this would be like sea stars, or what when I was a kid we called starfish, but they're not fish, so they've since started calling them sea stars. Sea stars and sea urchins are kind of in this group here. Let's see, what else do I have on here? The annelids here. These are like filter feeders. So lancelets are, the annelids are earthworms and leeches. These Amphioxus group, I'm not a taxonomy guy, so pardon my misspeaking in this as well. So the Amphioxus group, that is like filter feeders and lancelets. The annelids are earthworms and leeches. The tunicates are like sea squirts. So these are all, as you can see, they're kind of like not very, I don't want to say complex. They're complex organisms, but they're not like, have a lot of anatomy complexity, right? So they don't have like a lot of different organs and things like that. Like bivalves and things like that aren't super complex in that way. They may have complex genomes or complex physiology, but like their structures are not very complex. And then these already, you kind of got an idea already of what these groups are. So that is it for this quick cleavage tutorial. We'll go into a little more cleavage in the next one, and stay tuned.